Hello everyone. Welcome back to Microbial Engineering and Algal Biotechnology Laboratory MeEP Sciences channel. I, Dr. Dipankar Ghosh, Assistant Professor, Department of Biosciences, JIS University, Kolkata, will deliver a talk on evolution of SARS-CoV-2 variant, a glimpse of scientific intervention. First of all, I would like to acknowledge to all of my MeEP roomies, especially the summer internship student of 2021 session, Shonchoita, Shucheta, and Polomi for their immense and great support to make this particular presentation available online. So let's get started with today's discussion on the SARS-CoV-2, a specific scientific intervention. So these are the content that we are going to sequentially going to deliver to all of you to make you understand in a brief outline, followed by the basic idea about SARS-CoV-2, how they got originated, and how does they got mutation, and they will carry out the infection in human, other animals, and how the different variants of SARS-CoV-2 has been evolved. What are the different variants of concern, and what are the different types of mutations are got introduced in their amino acid level that we are going to discuss. Following the process of tracking of different mutations and the variation events, and we have to also like to special emphasis on why is it crucial to comprehend the evolution of SARS-CoV-2, followed up by the future outlook. So let's get started the discussion of today. What is SARS-CoV-2? Severe acute respiratory syndrome coronavirus 2 is a virus that causes COVID-19, which is a respiration illness, also known simply as a coronavirus. It was previously referred by the provisional name 2019 novel coronavirus, has also been called human coronavirus 2019. So SARS-CoV-2 is a positive sense single-stranded RNA virus that is highly contagious in human. As described by National Institute of Health, it is very successor to the SARS-CoV-1, the virus that caused 22 to 2004 SARS outbreak. The virus can spread from an infected individual mouth or nose in the small liquid particle or droplets when they cough, sneeze, speak, sing, or breathe. So these particles ranges from larger respiratory droplets to the smaller aerosols. So what is the basic concept and the background on origin of SARS-CoV-2? The first known infection from SARS-CoV-2 were discovered in Wuhan, China. And many of the early infectees were workers in the Wuhan seafood market. It has been suggested that the virus might have been originated from the market, but there was no clear indication for that. However, other research indicates that the visitor may have introduced the virus to the market, which then facilitated rapid expansion of the infection. The March 2021, the WHO conceived the concept and report that have been stated that the human spillover via an intermediate animal host was the most likely explanation, with the direct spillover from the bat next most likely. So, introduction through the food supply chain and the human seafood market was considered another possible way out, but less likely explanation required. Research into the natural reservoir of the virion particle that caused 2002 and 2004 SARS outbreak has resulted in the discovery of many SARS-like bat coronaviruses. So usually the phylogenetic analysis indicates that the samples taken from the Rhinolophus sinensis shows a resemblance of the 80% of the SARS-CoV-2 variant. Phylogenetic analysis also indicates that the virus from the Rhinolophus affinis collected in the Yunnan province, designated also as rat TG13, has a 96% similarity to SARS-CoV-2. Bats are usually considered the most likely natural reservoir of the SARS-CoV-2. Differences between the bat coronavirus and the SARS-CoV-2 suggest that the human may have been infected via the intermediate host. Although the role of pangolin as an intermediate host was initially positioned, and that has been published July 2020 as suggesting that the pangolin are the intermediate host of the SARS-CoV-2 like coronaviruses. Bats are most likely considered the natural reservoir, although the role of the pangolin as an intermediate host was initially positioned. Evidence that The hypothesis includes the fact that the pangolin virus sample are two distal 2019 SARS-CoV-2 isolates obtained from pangolin seized in the Guangzhou were only 92% identical in the sequence of the SARS-CoV-2 genome. That the recent consequence studies have been revealing these particular facts. So the evidence against this hypothesis includes the fact that the pangolin virus samples are too distant to SARS-CoV-2. Isolates that have been obtained from the pangolin seized were only 92% identical in sequence to SARS-CoV-2 genome. In addition, despite the similarities, resemblance in a few critical amino acid residues, the pangolin virus sample exhibit poor binding to the human ACE2 receptor. Now the question comes, how does SARS-CoV-2 mutate and when it infects human beings? When a virus replicates or makes copies of itself, it's sometimes altered a little bit. These changes are called mutations. The virus with one or several new mutations is referred to as the variant of original virus. So the more viruses circulate, and they have highly prone to may get altered in their genotypic level. So these alterations can occasionally result in a virus variant that is better adopted in its environment compared to the original virus. This process of alterations and selection of successful variants is called the viral evolution. Some mutations can lead to changes in the viral characteristics in the salient features, such as altered transmission. For example, it may spread more easily and or more severity. For example, it may cause more severe disease and the progression. Some viruses change quickly and others are very slowly. SARS-CoV-2, the virus which causes COVID-19, that tends to change more slowly than other such HIV or influenza viruses. Every time SARS-CoV-2, the virus that causes COVID-19, they do infect someone, it picks up tiny alterations in its genetic code as it makes copies of itself. Most of these mutations do not lead to anything. They are evolutionary dead ends. Others don't alter the virus's behavior, but are also not harmful to it. If these can spread to other people, they can form new variants of variant particle. There are some suggestions that variant may have emerged in people suffering long-term infection. Unable to fully clear the virus from their bodies, their infection can last several months in ongoing battle with their immune system or the treatment they receive. There are also indications that new variant could emerge as the genome of two different COVID-19 viruses get mixed, shuffled together while infecting the same person, an event known as the recombination phenomenon. The apparent cause of the severe form of the virus is due to the reassortment event. The influenza variant is made of independent segments that may reassemble or the reassert upon co-infection with similar virus. Interestingly, these highly virulent variant variants did not involve the hemagglutinin or neuraminidase protein that are required for virus to infect and be released from the host cell. These underscore again it is crucial to consider the viral features beyond the protein that are readily recognized by the adaptive immune system. The SARS-CoV-2 does not reassort because it made of a single RNA element, but it is prone to recombine with available RNA intermediate. As a consequence, the most worried evolutionary features of the virus is the short term and is that coronavirus may undergo extensive recombination. The SARS-CoV-2, the control of this process entirely depends on the activity of the protein NSP14. The recombination can naturally stem from the co-infection with different strains of the same virus. And in this respect, this could set the Muller's ranchet as does sexual reproduction and more generally horizontal gene transfer. But it can also use RNA from other viruses or even artificial constructs. The SARS-CoV-2 spray primarily through human-to-human transmission, but there is an evidence of transmission between human and animal. That is the indication of zoonotic nature of this particular virus. There have been reports of large animal outbreaks in the mink farm in several countries. The SARS-CoV-2 can alter while infecting minks. It has been observed that this mink variant are able to transmit back into human through the close contact with the mink again. And the preliminary results have been suggesting that the mink variant infecting human appear to have the same salient features as other variant of the SARS-CoV-2 had. further research. That's why it is very requirable to better understand or to get better comprehension whether this Ming variant will cause sustained transmission among the human, and that could also have a negative impact on the countermeasures as a vaccine. WHO works closely with other organizations, research institutions such as the Food and the Agriculture Organization of the United Nations and the World Organization of Animal Health to evaluate the situation of the SARS-CoV-2 in animal and transmission occurring between the animals and humans. Then what are the different kinds of mutation they do refer in SARS-CoV-2, the novel coronavirus? There have been a number of missense mutations have been observed on SARS-CoV-2 genome. That is N440K. The name of mutation is N4404, refers to an exchange whereas asparagine is replaced by lysine residue at position of 440. Similar way, L452 R E484Q, E484K, N501Y, P681H. They are also the different terms of mutations in the protein level in SARS-CoV-2. Then what is the major concern that lies behind if the new SARS-CoV variant has been evolved due to the infection and emergence and reemergence through the reinfection process? WHO with partners are currently tracking these virus variants of the concern that are circulating around the globe. The first B117 that was first identified in United Kingdom, the second B1351 that was first identified in South Africa, and the third P1 which was first and it's circulating in Brazil in Southern America, but it was identified among the travelers arriving in Japan. So the researchers are tracking changes in the virus, what it means in terms of transmission, severity, impact, or the potential impact on diagnostic applications, therapeutics, and development of vaccine, so far and so forth. That's why the information that we have that there is increased ameliorated transmittability in B117 and B1351 viral variants. And this is resulting from the mutation, the evolution that allows the virion particle to become variant to be able to bind to the human cell and more easily and more having more accessibility. We do not see an alleviate yet in transmission with P1, but this is currently under investigations. Researchers have been working on it in terms of severity, and there are several studies from the United Kingdom that suggest that B117 have the increased severity compared to P1. There are studies that are underway that are evaluating the effort of the vaccine against these viral variants. And from the information that we have so far, the vaccine still work against these virus variants. Exactly, but how this variant emerged in still is a puzzle. All three carries clusters of mutation. B117 has 17 mutations on its spike protein. For example, while B1351 has 8 distinctive mutations on the spike protein, it is quite unusual to get large clusters of mutation like this. That's why it's probably never know exactly how the new variant of concern first have been emerged. But we are now able to watch them as they are spreading, remerging as they continue to alter. Already we have spotted a handful sequences of B117 variant in United Kingdom and that have been picked up a mutation known as E484K. Then what is the exact process, the way out of tracking the different mutations and the variant that has been caused on the SARS-CoV-2 genome as an evolutionary prospect? The research groups in all over the world have carried out genome sequencing of the COVID-19 virus. The genomic sequencing is basically the avenue of determining the entirety or nearly the entirety of the DNA sequence of an organism's genome at a single time. After sequencing, researchers shared these sequences on public repository domain as a database, including GISAID. This global collaboration allows the scientists to better track down how the virion is changing. WHO recommends that all the countries ameliorate the sequencing of COVID-19 virus where possible, share data, big data set to help one another monitor and respond to the evolving pandemic. WHO has also established the SARS-CoV-2 risk monitoring and evolution or evaluation on the framework to identify, monitor, access the variants of concern. It will not only involve the component like surveillance, research on variants of concern, they also evaluate the impact of diagnosis, development of the vaccine and therapeutics against the novel SARS-CoV-2. The framework will serve as a guideline for manufacturer for the countries on changes that may need it for the COVID-19 vaccine development. During late 2020, the emergence of variants that posed an increased risk of global public health prompted the characteristics and characterization of specific variants, which are variants of interest, that is called VOIS, the variant of concern, VOC, in order to prioritize the global tracking and research undergone through ultimately to inform the ongoing responses to the COVID-19 pandemic. So the researchers have been monitoring and they are reassessing the evolution of the SARS-CoV-2 since January 2020. The established nomenclature system for raming and tracking SARS-CoV-2 genomic lineage are being maintained by GISAID. Then why it is so crucial, so important, so primordial to understand the evolution of the SARS-CoV-2? These evolutionary studies can review the phylogenetic analysis on SARS-CoV-2 variant and different variants to classify them to find out their possible ancestor and the offspring. The current researchers are trying to provide a wide range of different coronaviral genus, lineage, strains, isolate for a better understanding of the molecular epidemiology of the virus that will allow to understand how they are getting transmitted, how the retransmission or emergence will actually induces the severity or the efficacy of the SARS-CoV-2 virus that will make more dramatic and more enhanced elevated form and efficacy of infection. Further studies for exact receptor like ACE2 receptor or TREMPRESS2 and other virological features on the surface of spike proteins also should be suggested. Possible vaccine strategies, effective treatment can be possible by better understanding of the virus possible and to find out the novel pathogenesis, their molecular mechanism that will allow you to identify the targets for repurposing or the development of new therapeutics or the development of the vaccine. Effective drug design also should be possible by understanding the entire metabolic pathway and different molecular mechanism, signal cascades, those who are directly, indirectly associated with evolution of the SARS-CoV-2. So now we come to an end of this particular lecture session. So we will try to wrap up the entire session by our own opinion and thought process of different researcher as a future outlook. So COVID-19 vaccine that are currently available or in the development are expected to provide at least some protection against the new or novel viral variants because these vaccines elicit a broad immune response involving a range of antibodies and cells. Therefore, changes or mutations in the variant particle should not make vaccine completely ineffective or least effective. In the event that any one of these vaccines prove to be less effective against one or more variants, but there is no clear-cut proof for that, it will be possibly to alter the composition of the vaccine or the formulations of the anti-therapeutics for repurposing purpose to protect against these variants. The data continues to be collected and analyzed on new variants on COVID-19 virus to make the drugs or the therapeutics in much more potentiality. While we are learning more, we need to do everything possible to cease the spread of the virus in order to prevent mutations that may reduce the efficacy of existing vaccines and the repurposed drugs. In addition, the manufacturers and the programs using the vaccine may have to adjust to the evolution of the COVID-19 novel coronavirus. For example, vaccine may need to incorporate more than one strain or isolate when in development. Booster shots may be required, and another vaccine changes may be required as well. According to the CDC, recent studies show that all these three COVID-19 vaccines organized for use in United States, like Pfizer, Moderna, Johnson & Johnson, they work against COVID-19 viral variants, including the Delta strain. So people who are unvaccinated at a risk of getting serious sick from the Delta virus. So it's always be a good idea to get vaccinated as per the government rules, as per the nomenclature provided by or the standard operating protocol provided by the government authorities. But it is possible for vaccinated person to test positive for Delta variant of COVID-19 because it is also very true that no vaccine is 100% effective, known as a breakthrough cases. The cases in vaccinated individuals are usually mild, but they do not get died of. They are spread, but the severity of spreadation will be less. Even they don't feel sick when they got vaccinated, rather than not being unvaccinated. So this does not mean that the COVID-19 vaccine don't work. The vaccine are still doing what they are supposed to, protecting people from getting serious illness. So that's why the vaccine that we have been using, they are none other than the immune booster. So it is always be a good idea to take vaccine, not to remain unvaccinated. Then you will reduce the chances to get hospitalized and dying due to COVID-19. So these are the basic references that we have been read, we have utilized to prepare the entire session. And if you want, you can go for further studies by downloading those papers, and you will get more information. You can enlighten yourself. Thank you very much. So this is my current research group where PhD scholars have been working, and also MSc research trainees are there currently engaged. Thank you very much for listening this session. I hope you have enjoyed and you have get a lot of knowledge in a very summarized way. Thank you very much for supporting our channel and also supporting our research group at GIS University, Kolkata Department of Biosciences. Thank you very much.